Okay, hello, this is chapter 2 of Edexcel Chemistry, and we are talking here about experimental techniques. I have compiled in this chapter all the information, or almost all the information that you need um, for any questions regarding experiments. So let's take a look at this. The first thing that you need to know is the name of the apparatus. Of course, apparatus means whatever we use in the lab. So you need to know the names of all the things that we use in the lab. This is basic. What is the name of this? This is something that we use every day in the lab. This is test tubes. What's the name of this one? This is a conical flask. Okay? Sometimes you can just say flask. That's fine. But it's actually a conical flask. This one is also a flask, but this is another form of flask, so we call it a round-bottomed flask. You could also just refer to it or label it as flask. What's this? You should know this is a beaker, and please be careful with spelling. When you're studying all of these things, you are expected to write the correct spelling, so please notice the spelling of beaker. What are these things in the container? These are things that we use to stir solutions. These are called glass rods. What about this one? You should know this one. This one is for filtering. This is a funnel. What are these? This is something that we use to crush solids. We call it mortar and pestle. So the container is the mortar, and the handle that you use to crush the solid, that is called a pestle. What's this? It looks like a glass container. Some people mistake it for lenses that you study in physics, but this is not actually a lens. This is called a watch glass. It's just a piece of glass that we use just as a container for solids, for example. Okay, so usually in this course they will mention it when we're talking about heating a liquid that evaporates easily, which we call volatile liquid. If you're heating a liquid like ethanol, which evaporates easily, you will probably want to put a watch glass on top of the beaker. This is to avoid evaporation of the liquid. What's this? It's actually a small container with a cover, and we use it to heat something like solids or something that gives out a vapor. So this is called a crucible and its lid. Okay, this is something else that we use to heat solutions in. This is called evaporating dish. This, of course, is what? This is a sensitive balance. And what's the name of this? Please don't call it a heater. Don't call it, I don't know, fire. It's a Bunsen burner. And again, please note the spelling. So this is a Bunsen burner. This is something that we use to add drops. So if I say I'm adding 3 drops of a liquid to something, then I'm using this, and that is a dropper. What's this? This is used to measure volumes of liquids. This is called a measuring cylinder. It is used to measure volumes of liquid if I don't really need them to be very accurate. So if he's saying add about 50 centimeter cubed of a liquid, or about 30 centimeter cubed of a liquid, or if he's saying add 25 centimeter cubed, but he did not say 25.0, that means he doesn't really need it very accurately. Then I'll probably need a measuring cylinder. But if I want to measure the volume of a liquid accurately, then I should use this. This is something that has graduations, and it has a tap. Now, something with graduations and a tap is called a burette. Again, please notice the spelling. Okay, so this is an accurate method of measuring volumes, and it can be used for any type of volume. So it can say 20.0, 20.1, 20.2, 20.3, 21.5, whatever. So any volume of liquids, you can use a burette. If you need to measure volume of a liquid accurately, but it is something specific, for example, this one measures exactly 25 centimeter cubed of a liquid. It cannot measure more or less. It can measure multiples of 25. So if I need 50 accurately, then I can use this twice. Or if I need 75.0 centimeter cubed, I can use this 3 times. But I cannot use it to measure, for example, 20 or 10 or 30 or whatever. So this is an accurate method of measuring the volume, but specific volume. So this is called a pipette. Remember, it doesn't have a graduation. It doesn't have graduations. It just has a mark that shows you if you fill it to that mark, then it is exactly 25. It also does not have a cap. So this is a pipette. Okay, this is something that we put our beaker on or our flask on in order to heat it. So this is called a tripod stand. Now, if I want to use my burette, I need to hold it upright somehow. I use the stand to hold it upright. It is attached to another thing that is attached to the stand, and that other thing is called the clamp. So the thing that's actually attached to the burette is called the clamp. That tall apparatus that is holding the burette, that's called the stand. This is used to measure temperature. Of course, this is thermometer. This is used to take solids, to just scoop out solids out of its container. So if we say add a solid to my beaker or add some solid to my beaker, I use this. This is called what? It's called a spatula. Okay, this is used to measure volume of gases, and it's an accurate method. So if he says collect a gas and measure its volume, then you need a gas syringe. So if I have a reaction mixture in my flask, it is giving out a gas and I want to measure the volume of gas, then I use a gas syringe. This is used to hold something that's hot. This is called tongs. Okay, so we use it to hold a hot beaker, a hot crucible. This one is used to hold the hot test tube. So this is a test tube holder. Okay, this is what we call a condenser. We will talk about it again when we're talking about collecting liquid. If we're doing distillation, this is a process of distillation in which you heat a liquid, it evaporates, and then it is cooled and condensed. So this uses the condenser. We will talk about that later. This is just a tube going from one place to another. So this is called a delivery tube. So I have something going from my flask to that other test tube. Then I connect them with a delivery tube. Now, this is something that is similar to the measuring cylinder, but it doesn't have any graduations. So if it is similar to a measuring cylinder, but no graduations, it's called a gas jar. Now, what is this? These are two types of funnels that we put on top of flasks. If it doesn't have any graduations, no tap, it's called a thistle funnel. If it has no graduations but it has a tap, it's called dropping funnel. Remember that this is different from a burette. The burette has a tap, but it must have graduations. This one is a dropping funnel. So the dropping funnel or the thistle funnel is something that you put on top of the flask in order to add a liquid to that flask during the reaction. Okay, this was the clamp that we said is used to hold things like burette, or it can be used to hold any other apparatus. Now, to put the flask on a tripod stand and heat it, you need to put it on a wire gauze. So that thing below the flask is a wire gauze. This is a tube that is U-shaped, so they actually call it a U-tube. So this connects, if I have a gas and I need to pass it through some sort of solid in order to dry it or to react with it or something, then we use a U-tube. Okay, safety precautions in the lab. What should I do to make a reaction safer? Now, first thing, if he's asking you to mention a safety precaution, then you look at what you're doing. What are we doing? If you find that in the experiment we're heating flammable liquids, so ethanol for example, if we're heating something that is flammable, it can catch fire, then I have to heat it in a hot water bath. And we said a hot water bath means you just put it into a beaker containing water, and this is what you're heating. You're not heating the test tube directly. We cannot put ethanol near an open flame, again, because this is flammable. If the experiment produces gases, so I look at the experiment. If it is something that is flammable, I say heat it in a hot water bath. But what if I'm not using ethanol, for example, I'm not using something flammable, then what should we do? We look again and see if it is an experiment that involves gases, whether it is using gas to react or it is something that's giving out a gas, then I tell him do the experiment in a fume cupboard. A fume cupboard is a huge thing with a door, a sort of small door that you can lift up, put your things in, and then close it a little bit so that when you're working with the experiment, you do not breathe in the gases that are being used or that are being formed in the experiment. So that's called a fume cupboard. Now, if I'm not doing anything with gases, then I need to tell him hold the hot objects with tongues. So if I'm not using gases, I'm not using ethanol, I'm heating it, then I can tell him hold it with tongues, or wear safety glasses to avoid splashes in the eyes, or wear gloves when handling corrosive substances. So if we're talking about things involving acids or bases, then I tell him wear gloves. Okay, to improve accuracy of results. So if he says, how can I improve the accuracy of the results of this experiment? Okay, a basic thing would be tell him repeat each reading 3 times and obtain the average, for example, because that would be a more accurate result. Or if we're measuring volumes of liquids, then I tell him measure the volume of liquid using a burette instead of a measuring cylinder because it is more accurate. We said a burette is an accurate method of measuring the volume of liquid. So if he says that he used a measuring cylinder, then I tell him no, don't use a measuring cylinder, we should be using a burette. If he's talking about volumes of gases, then I will tell him measure the volume of gas using a gas syringe because it's more accurate. So measuring the volume of gases, you need a gas syringe. So these are all possible suggestions of improving accuracy of results depending on what the experiment is talking about. Okay, if we're drying a solid, he wants a pure dry solid. Usually, how do we dry a solid in the lab? The easiest thing would be dry between 2 filter papers. So if I have a solid I'm collecting or I'm preparing and I want it dry, I dry it between filter papers. This is important so that I do not break down the crystals, or I do not lose something like water that's supposed to be in water of crystallization that's supposed to be in the crystals and so on. Or I can tell him place it in a desiccator. A desiccator is a glass container with a cover and it's containing a chemical at the bottom. This chemical at the bottom is a drying agent. So something like anhydrous calcium chloride. And remember, if he's using anhydrous calcium chloride somewhere and he asks you why are we using it, then we're probably using it to dry the solid. Okay. Of course, I cannot heat crystals in an oven. Usually I will not. The only thing I can heat in an oven is if I'm trying to dry iron nails, for example. We'll be talking about experiments dealing with iron nails. Then I can dry that in an oven. But I do not put crystals into an oven because this may cause breakdown of the substance or decomposition of the substance, or loss of what we call water of crystallization. So if I'm trying to prepare something that is a hydrated solid, and we'll talk about that much more later, if I'm trying to dry a hydrated solid, I don't want to lose the water of crystallization, so I do not heat it in and out. Okay, hazard labels. Usually this is not very common as a question, but you should be familiar with these labels that are usually on the bottles of chemicals. The most common one would be the toxic one. Can you see which one means toxic? The one that has a skull means toxic. The one that has fire is flammable. The one that has a hand being affected by the liquid, that means that this liquid is corrosive. And remember that we said corrosive means it burns the hand. And just take a look at the others. Just be familiar with what this means. Okay? Okay. Usually you will be required to draw a graph. When you draw a graph, you plot the points and then you join. And before you join, please check the question and see if he's telling you to join it in a specific way. Usually it is just a curve. But if you're drawing a curve, it should be a smooth curve. So if he has a point that is away from the curve, which we call anomalous result, this anomalous result should not be included in your graph. So your graph should not be something that goes in and out like that next one. It should be a smooth curve like the first curve that we have here. Okay? If we're testing solids, a solid that is colored is something that has a transition metal. So remember, we'll talk again about transition metals. Transition metals are the ones that are found in the middle of the periodic table. Now, if a metal is a transition metal, then its compound would be colored. So if the solid is colored, that means it has a transition metal. So all of these have colors. That means they are made up of something that has a transition metal. It's a compound that has a transition, copper something, iron something, chromium something, then these would be colored. But if it's a white solid, then it doesn't have any transition metal. So as a solid, it would be white. But if I dissolve it in solution, we don't say the solution is white or the solution is clear. We say that the solution is colorless. So if something has no transition metal, then as a solid, it's white. If I dissolve it in water, then it is a colorless solution. When we say that a solid was heated and condensation forms at the top of the tube, so I'm heating a solid and I see something forming at the top of the tube, that means that that solid must have had water of crystallization, and that is called a hydrated solid. So keep this in mind as we go along. We will be talking about it again as the questions come along. Okay, how do we collect the gases? We said the easiest way to collect a gas is over water. So this is how we usually collect a gas. So this is an experiment that is giving out a gas. I have a delivery tube going into a big beaker, which we call a trough. And remember that a trough is just a wide beaker. And the gas jar that is on top of it is originally filled with water, but as the gas is formed in the experiment, it will be formed at the top of the gas jar and it will displace the water downwards. So this is called collection of gas over water. Now when do we do this? We do this if the gas is not soluble in water, if it does not dissolve in water. So remember that if I'm preparing a gas like oxygen or hydrogen, oxygen and hydrogen are not regarded as soluble in water, so we can collect them in this way, over water. But what if the gas dissolves in water? If it dissolves in water, you cannot use this. Then I have to collect it in another way. And that depends on whether it is lighter than air or it is heavier than air. So if we're talking about something like ammonia, ammonia is lighter than air. So I have to collect it by what we call upward delivery. So this is a gas jar that is upside down in that way. And what happens is the gas is lighter than air, so the gas goes up and it pushes the air that is originally in the gas jar down. So this is called upward delivery. If the gas is heavier than air, then we do it by what we call downward delivery. So it is in this position and the gas is denser than air, such as what? Such as carbon dioxide. So you have to remember that carbon dioxide is a gas that's heavier than air or more dense than air, while ammonia is lighter than air. So the carbon dioxide can be collected by downward delivery because it's heavier than air. And remember that we cannot collect it over water because carbon dioxide would dissolve in water. Now, if I'm trying to collect it and measure its volume, then I cannot use the previous methods. I have to use a gas syringe. So the gas syringe will take the gas and give you the volume. In some cases, we say we use mineral wool and we're heating this liquid. Now the delivery tube is going into water, so the delivery tube, if I stop the heating, this delivery tube will allow the water to suck back into the tube that I was heating. So a precaution in this case would be, if I'm trying to stop heating, I shouldn't stop heating with the delivery tube still inside the water. I have to remove the delivery tube first, then I stop the heating in order to prevent the back suction of water into that hot tube, which would break it. Okay, again, keep all of this in mind, and as we go along, I just compiled all the things that you need for your experimental methods, and then as we go along, we'll be repeating this. What do we use cotton wool for? We can use it for one of three things: either to hold liquids, so for example in this experiment we were trying to hold a liquid and the test tube is horizontal. If I just put the liquid in there, the liquid will fall out. So I put it soaked in mineral wool or cotton wool. These are the same thing. And in this case, the function of the mineral wool or the cotton wool is to hold the liquid. Or I can use it to prevent solids from passing through the apparatus. So for example, this is an apparatus. I have solids in them. I don't have a liquid or anything, so I have a solid. Why do I have a piece of cotton in there? This is just to prevent any of these solids from going through the apparatus into whatever I'm collecting. A third use of the cotton wool is to prevent loss of liquid by splashing. So for example, I have a flask on top of a balance, and this flask has something that is giving out bubbles of gas. If I just leave it open, then the bubbles or the liquid will splash while the bubbles are coming out. So the function of the cotton wool in this case is to prevent loss of liquid by splashing. A suction pump is used in some experiments, and this is to suck gases through the apparatus. So if I'm burning something and I want the gases coming out from the burning to go through the apparatus into that tube containing the liquid, then I can use a suction pump. Airlock is used when we're doing experiments on fermentation. So fermentation is just yeast and glucose, and it's giving out carbon dioxide. So this is something that's giving out a gas. So I cannot close it completely because I need the carbon dioxide to escape. But at the same time, fermentation is something that does not want oxygen or air to go in. So I use what we call an airlock. So the airlock is just a bent tube, as you can see, and it just has water in it. And this, in this case, it will allow the carbon dioxide to escape because gas bubbling out will just bubble through the water and go out, but it does not allow any oxygen to go in. So this is called an airlock. Okay, so when we measure temperature of experiments, some temperatures will go up and some temperatures will go down, and we will know that, or if you remember from last year, we will know that this is exothermic and endothermic reactions. But the thing is, if we're measuring temperatures, they will initially go down or initially go up until the reaction finishes. Now, what happens when the reaction finishes? Whenever the reaction finishes, then the temperature of the reaction will start to go back to what it started with, or the room temperature. So if he says that the experiment, the temperature went down, and then why did it go up again? Or the opposite, that is because the reaction has finished and it is returning to room temperature. Okay. When doing an experiment involving measurement of temperature, usually we do that in a polystyrene cup, not in a beaker. This is because polystyrene is an insulator, and an insulator means it does not allow loss of heat to the environment. So I'm trying to measure, this is an experiment, for example, that is giving out heat, and I'm trying to measure the temperature. Then I should do this in a polystyrene cup. Why? To avoid loss of heat to the environment, so that the temperature measures the accurate temperature. Thermometer, sorry, measures the accurate temperature. Okay, sometimes he might ask you to read a stopwatch. A stopwatch is just something that has minutes and seconds, and he will always tell you that the circle inside indicates minutes and the circle outside indicates seconds. Usually you're required to read the time in seconds. So this, for example, is 1 minute and something seconds. Then I can add them all up. A minute is 60 seconds. Add it to the seconds, which are what? This is 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25. This is 26 seconds. So you add the 60 to the 26, and that is what? 86 seconds. Okay, so that ends our lesson on experimental techniques. And what you're supposed to do now is go to the PDF version of the notes, sit down and study them, then go to the questions, sit down and do them, and in the next lesson we'll discuss the questions and their correct answers. Okay, try to study chapter by chapter. Please do not leave everything to the last minute. So if you study chapter by chapter, do the questions chapter by chapter, then you should be okay. Okay, so see you in the next lesson. Thank you.